First, gene-edited snakes use mysterious Turing patterns to achieve near-perfect hexagonal scales. Scientists used CRISPR editing to make the world's first genetically modified snakes, giving new insight into how the reptiles develop their patterned scales for the first time ever. Scientists have created genetically modified snakes. The CRISPR-edited reptiles are providing new insight into how corn snakes, Pantherophis gutidus, develop their precisely patterned scales. Much like feathers on birds or hairs on mammals, snake scales are the result of placodes, small, thickened structures on the skin that develop at the embryonic level, according to a new study published Wednesday, June 14, in the journal Sciences Advances. But unlike most other species including mice, where the placodes are random, a snake's placodes develop in a highly organized fashion, laying out the positioning of every single scale. Rather, the spatial organization of these placodes follows a pattern in nature first explained by mathematician Alan Turing, the researchers added. Scientists from Geneva wanted to know exactly how and why these near-perfect hexagonal patterns developed on the dorsal scales located on the snake's backs and flanks, but not on the ventral scales that form as a single row on the animal's underbellies. The researchers found that an embryo's ventral scales develop first and align with the position of somites, blocks of cells that determine the location of the vertebrae, ribs, muscles and dermis of the skin. Once the ventral scales are established, two separate waves of placodes develop, traveling toward each other. The waves meet laterally, creating the tidy hexagonal patterns that are the hallmark of a snake's skin, according to a statement. To confirm our work, we used computer simulations and received similar results. Lead author Athanasia Zika, a postdoctoral fellow in the Department of Genetics and Evolution at the University of Geneva, told Live Science. This is surprising because the pathway is essential for proper development of skin appendages in birds, reptiles and mammals. Zika pointed to lizards with a mutated ETA gene, which were previously studied in her university's lab, as an example of a reptile that never developed scales. This led researchers to create the world's first genetically modified snakes. Using CRISPR Cas9, which edits genes by severing the DNA and letting the natural DNA repair itself, Zika and her team successfully created mutant snakes that lacked dorsal lateral, hexagonal scales, but still had ventral scales. She said that this proved that the scales aren't self-organizing and occur without a functional canonical ETA pathway. In total, the scientists created 4 corn snakes, all of which are currently 2 years of age and are doing well, Zika said. The animals we produced are exactly the same as the naturally occurring snakes. We were able to reproduce the same phenotype, Zika said. She said they plan to conduct another round of CRISPR edits on the genetically modified snakes in 2 years, once they reach sexual maturity, to see if the mutation will transmit to the next generation.